Welcome back to the course Fundamentals of General Immunology. So today we'll discuss about the selection of the B and T cell population. What are the theories behind the selection of T and B cells? We'll try to look into in detail. So this is our lecture number 17, selection, editing and developmental tolerance. So let's try to understand the selection. So there are many theories which are put forth by various scientists, and some of them are well documented in terms of their originality, and also to the extent that these are replicated to the actual theory which we know to date. So the first theory is the side chain theory. The side chain theory was given by Paul Ehrlich in 1900. It was kind of a fundamental model to explain the antibody production, which we are following today. And it proposed that the cells have many receptors. So suppose if this is a cell and these cells express certain kind of a receptor, which we call side chain, and they can lock with an antigen. So suppose if antigen is coming which are cognate to this receptor, it will lock the antigen into that site. So this receptor is what is very important, because that can do bind to this antigen. And if there is a binding of an antigen, so it says if there is an antigen binds, the cell produces an excess of receptor, releasing them as an antibody to neutralize the threat. That means once these are recognized, then particular cell type will express more of these molecules, and these can be released in the system. For example, if it is in the body fluid, in the plasma or any serum, to neutralize these antigens. So essentially, once there is a recognition of an antigen with this receptor, the receptor can be produced in a large quantity, and this receptors will be then solubilized into the body system in order to neutralize the antigen. And this was the fundamental theory which is valid to some extent today also. Gave 1908 a Nobel Prize of Physiology in Medicine to Paul Ehrlich. So this is the side chain theory, and thereafter there are many other theories which were proposed in order to show that how these B cells are selected, how these antibodies are produced, and why there are so many repertoires of antibody which can generate such a diversity of antibody in the human body when there is a multiple antigen coming inside the human body. So the second theory is, so let's try to understand the same side chain theory in a diagrammatic representation. So say these are the cells and these are all the receptors which are present, and some of the receptor can bind to these antigens which are coming from outside, and once these are identified, then more and more production of these receptors may happen in the cell, and then these can be solubilized into the body system in order to neutralize the antigen. So this is what exactly was the side chain theory, which says that the more receptors are produced in response to the entering antigen in order to neutralize them. So this is what the side chain theory is. And thereafter there are many other theories which were put forth by different scientists, and one of the next theory was given by Neil Jern in 1955. That was natural selection theory. Later on he also proposed the network theory, for which he got the Nobel Prize later. And the remarkable thing about the Jern theory was he predicted to some extent the idiotype and anti-idiotype antibodies, which can forms the literally the baseline for the ELISA or any kind of a diagnostic tool which we are developing now today. So the core principle of Jern theory was, so Jern proposed that the body produces a massive diverse pool of natural antibody during the fetal life. So what he proposed is, during the early age of the life, the pool of antibodies are produced. So these are not specific to antigen, these are just pool of antibodies which are produced at the very early age stage of life. When a foreign molecule or foreign antigen enters into the body, it selects the specific pre-existing antibody that best fit it. That means we have, suppose, a pool of 100 antibodies existing in a fetal life, and out of those 100, suppose these are fitting, 1 or 2 may fit to the antigen which are entering inside the body, and then based on that fit model, that particular cells are selected, and it will start stimulating the production of that similar kind of antibody to a larger extent. And these are actually based on the first model where the pool of the antibodies can select one of the antigen. So it is rather a specificity at the very early stage, and it's not depend upon the antigen as a template. So the antigens may not act as a template for the generation of antibody. It's simply that there are pool of antibodies which are present in the fetal life, and when there is an encounter of an antigen later on the life, some matching will be there, and once the match is there between the antigen and antibody, that particular cell type will proliferate to produce a more similar kind of antibody, and then we can see that this is the same will neutralize this antigen in future. So this was a core principle of natural selection theory, which later on, because Jern proposed another theory of network hypothesis or network theory, which says that the immune system is a functional network of antibodies, which he proposed in the first time also, and he named it idiotypes. And then there could be an anti-idiotype, that means an antibody, and the second one is anti-antibody, that means there will be a protein which can bind to another protein and which can regulate each other to maintain the balance. Now let's try to understand it in a diagrammatic way so that it is more clear. So the thing is, like, once there is an antigen, there is a production of antibody, and then there is an anti-antibody which is maintaining the network, and once this network is maintained, the pool of these antibody and anti-antibody can clear the infection in the human body. So let's see in this diagram the network theory. So suppose this is an antigen, and this antigen is expressing this epitope on the surface, and this is actually an antibody which is having a variable region and the constant region. And this is just a picture which Jern proposed at that time, which largely modified today. And then we could see that the variable domain, which is actually a paratope which we know today, can bind to this antigen. Now of course this is an cognate antibody that can bind to antigen, but there could be a production of a second line antibody which is an anti-antibody. So this could be against this particular antibody, which acts as a template to form another antibody, which looks very similar to this antibody. So what is proposed is that once there is an antigen present, the epitope of an antigen is identified by an antibody. There is a production of anti-antibody against the similar kind of antibody which recognizes the antigen, and this anti-antibody acts as a template to produce another antibody which is actually similar to the first one. That means if there is a pool of this antigen antibody reaction, then probably this particular antibody will neutralize this antigen when they are coming into the body system. So there is a network which works together. That is what Jern proposed at that time, while the one which is produced in a different style may not able to neutralize the antigen, but there could be a cognate production of another other kind of receptor in a pool of antibodies. So there could be a network of everything, and one which is closely fitting to the antigen will neutralize the antigen in future. So this is what is network theory which put forth by Jern. And later on, the most plausible theory was the clonal selection theory, which was given by Burnett in 1957. So this is classical theory which is valid till date, and that says that the clones are selected. Clones are generally the cell which are very similar to each other. Generally this is a term applicable to a bacterial cell population where one bacteria is very similar to the other bacteria post replication. So suppose there is a cell which is after division forms a two cell. This cell can be called as the clone of the first one. Their genotype and the phenotype are exactly the same. That means the production of this, suppose this produce A, this will also produce A, this will also produce A. If this is producing a similar kind of molecule, they are called as the clone cell types. So this is what the proposed theory was, and the core principle depends upon the 5 distinct set. One says that the diverse repertoire, so the immune system maintains a large pool of diverse repertoire. What does it mean? It means that the whole immune system is having a variety of lymphocytic and population. Now, in general, the human body contains around 10 to power 12 lymphocyte, which is approximately 3004 lower. So in human body, there are production of 10 to power 12 lymphocyte in general, while in case of mouse, it is approximately say 3004 lower as compared to human. So in the mice, you can see that somewhere around 10 to power 8 lymphocyte population is there, and in case of human, we could see that there are more number of lymphocyte. So what it stands for, there are a diverse pool of, we could see that there are many different kinds of lymphocyte which are present in the human body just to fight against the different kind of antigens which are entering inside the body. So diverse repertoire is the first one. Second one, pre-existing specificity. The second is pre-existing specificity. What does it means? That the lymphocyte are specific to an antigen before the antigen entered inside the body itself. That means once there is an entry of the antigen, of course it can bind and recognize that antibody, but there are diverse repertoire. So before entering even the antigen, even entering inside the body, we do have lymphocyte that can bind very specific to the antigen. So there is no prior kind of a knowledge required for that. There is already a pre-existing specificity that exists in case of lymphocyte. That third one is clonal selection and expansion. That means when there is a binding of a specific antigen to the diverse repertoire of antibody, the cell is recognizing the particular antigen, there is a receptor antigen interaction, and once there is an interaction, there is a selection of that particular cell type, and once there is a selection, the same cell population will be expanded to a larger content or quantity so as to produce a similar kind of receptor what we have understood in case of the first side chain theory. So say we can similarly see that the similar kind of cells will be clonally expanded in order to fight with the antigen. And once there is a fight with the specific kind of a repertoire, then some of the cells which can recognize those very effectively, right? These clones differentiate into effector cell. Of course, these are the secreting plasma cell, and some of the cells will go as a memory cell, so they can store all the information which can help them to fight against the future entry of the similar kind of antigen. So essentially it will allow a rapid kind of a development to a re-exposure of the same antigen, which is what is actually the vaccine biology which we learned today. So the memory response is also very important, because once the antigen is entering, one has to remember the type of antigen, category of antigen, and also the structure conformation of antigen, so that the antibody can be produced against very specifically the target to neutralize it. And finally, the self-tolerance. So suppose the lymphocyte that recognizes the body's own tissue is not safe for the body. Say if there is a self-antigen recognition, the body will produce an immune response against own cell population. So this is kind of leading to autoimmunity, and therefore these cells are actually eliminated by process of apoptosis. So this is what we call self-tolerance. So the core principle of clonal selection theory is, again, there is a diverse number of lymphocytes present in human population and also in other species as well. There is a pre-existing specificity, so that a lymphocyte can be identifying a particular kind of antigen even prior to the experience of the structure of the antigen. Then there will be a clonal selection and then expansion of the similar kind of cell. So one lymphocyte can produce a similar kind of antibody, that is what it stands for. And then post expansion, there could be an effector cells which can produce more and more antibody to destroy. And then there are some cells which convert themselves into memory cells, which can store the information of the antigen for the future generation when they are encountering the similar kind of antigen in future. And finally, the self-tolerance, where a self-reacting lymphocyte or that self-reacting antibody producing lymphocyte can be eliminated from the body system in order to avoid the self-immunity or autoimmune response. So these are the typical clonal selection theory, which was actually proposed by Burnett, for which he also got the Nobel Prize, and this is what is fundamentally we are following till today. So let's try to move on and see that what is the immune cell selections. We primarily know that there are two types of lymphocyte, T and B cell. So there is a maturation of the lymphocyte that happens because of the foreign pathogen, and it can produced in a large quantity. And there are two types of selection processes that are reported. One is called positive selection, which can produce an antibody, the cells which are selected to produce an antibody, which can react to the antigen. And the second is negative selection. So the negative selection stands for the deleting those self-reacting cells. That means we have to select the positive cells which can produce effective immune response, and we have to negatively select those cells which are reacting to our self-tissue. That means the definition wise, primarily T and B lymphocyte is a rigorous maturation process that ensure cells can recognize foreign pathogen while ignoring the body's own tissue. That means there should not be any self-reacting antibody that start destroying its own cell population. So those must be negatively selected, while the others which can be selected positively. Now the site for this selection is either thymus for the T cell and the bone marrow for B cell. So we could say that the thymus is the site for T cell selection and the bone marrow is for B cell, which is another subset of the lymphocyte. So we will see that how the T cell and B cell are selected in thymus and bone marrow respectively. So let's try to understand the T cell selection from thymus point of view. So the untrained thymocyte, so we can simply say that these are not expressing any receptor on the cell surface. We can simply say negative, negative. So these are thymocyte, means these are the cells of the thymus. So basically these are coming from the bone marrow, and so the untrained cells are entering into the cortex area. So the cortex is the central area and the outside we call medulla. So we'll see in detail about how this maturation process goes in thymus from the cortex to the medulla. So once the cells are entering, they are not trained, so they are just not expressing any cell type. And once there is, so once they are encountering with an MHC expressing proteins, these thymocytes are now successfully identifying these protein structure, and they undergo some kind of a modification. So in the cortex of the thymus, suppose these are maturing, these are encountering the MHC1 loaded structures from the thymic epithelial cell, then the thymocyte can express CD4 plus and CD8 plus cell, while the one which doesn't bind with the MHC will simply destroy by apoptosis. That means once there is a reaction of the thymocyte to the reactive MHC, then we see the expression of the antibodies are coming. If it is not coming, then it will undergo apoptosis. So once this is binding to the MHC, then only those things are going in the next step. So once there is a positive selection, this particular cells are continue to the double positive stage, and then it goes to the site where they can receive both the receptor CD4 plus and CD8 plus. Now we know that there are two types of receptor which are present in the T cell. One is called CD4 and the other is called CD8. And these two CD4 and CD8 receptors are expressed in two different T cell types. So what we have understood till now, that the cells are coming from bone marrow to thymus, and in thymus they are encountering the MHC1 loaded antigens presenting cell, and once they are identifying this antigen presenting cell, they are positively selected, and if they are not, then they are destroyed by apoptosis. Now since there is a double positive cell, that means CD4 and CD8 plus expressing cells are there in the thymus, then we could see in the next stage what is happening is there is a negative selection because there are double positive cells. So what will happen that and this process is happening in the medulla of the thymus. Now the earlier process were in the cortex in the middle part, now it is in the medulla of the thymus. So if the dendritic cell loaded antigen presenting cell is coming, and probably if there is no binding, so it should not bind to the self-antigen. That is what we are emphasizing again and again. So if it is binding to the self-antigen, the cell will be destroyed. And if it is not binding to the self-antigen, then the cell will survive. So here we can see that these are not identifying the self-antigen which are carried by the dendritic cell, so this will survive, and the one which can bind very specifically to the self-antigen will kind of destroy, because ultimately this can lead to the autoimmunity. Now once this is there, the reactivity in terms of the self-antigen, we can see that there could be a selection. The cells which are expressing the receptor and not identifying the self-antigen are going to the next step. And then the cells which are expressing two of the receptors, say cells are expressing CD4 and CD8 together, they will be destroyed. So the only cell which can express only one type of receptor will survive. For example, the cells which can express CD4 can survive to make helper T cell population, and the cells which can express CD8 will survive to produce a cytotoxic T cell population. So this differentiation of T cell will take place after the selection of the self-antigen reacting T cells. So we could see that once there is a production of naive T cell which is coming from thymus, then they are educated to identify the MHC1 molecule, they are selected. If they are identifying it, and if they are not, they are destroyed. And then next round is when they are identifying the self-antigen, then they are destroyed, and then when they are not identifying the self-antigen, then they are selected. So, and again in the final round is differentiation of the T cell, when both CD4 and CD8 expressing T cells are segregated separately. The cells only expressing one type of receptor can survive and go into the next round of process. So let's see the other aspect where we can learn the B cell selection. So same like T cell selection, the B cell are also selected, but the location is bone marrow and the peripheral lymphoid organs. So these are critically a B cell population. We could look into the CD4 and CD8 in the T cell population. Similarly, in the B cell population, there are a few terms which we need to understand. So again, the B cells which are reacting to the self-antigen have to be destroyed. So these are negatively selected, and these are called tolerance. So there could be two levels of tolerance. One is called central tolerance, which is where the immature B cell that is strongly bind to the self-antigen undergo apoptosis, either by clonal deletion or by receptor editing, which we will see in the next class. But typically there could be a removal of the cells which are reacting to the self-antigen. And if at all there are cells which can escape this central tolerance, can go to the periphery, and these cells are again tested before going to the actual site of action, and there also there could be a negative selection which is called the peripheral tolerance. So it's cell escaping the marrow are tested again in the periphery to ensure they do not become autoimmune. That means they should not produce an immune response against the self-antigen. So there are two ways tolerance is working in case of B cell. One is central tolerance, and then the second is in the tissue level, which is called peripheral tolerance. The positive selection is basically the term which can allow the cells to grow more because the receptor can identify the antigen effectively, so those cells are selected positively. And then there is the germinal center selection, where the mature B cell upon exposure to the foreign antigen in peripheral lymphoid organs undergoes further selection to maximize affinity for that antigen. That means, so there are two ways the tolerance is working, central and peripheral. And then finally there is a positive selection. Once there is a positive selection, then again we need to enhance that same cell which can bound, which can produce more and more effective cell. For that we need a germinal center selection, where the mature B cell population is enhanced in production in order to raise more and more antibody. So this is the B cell selection. We'll see it in details in this slide now. So let's try to understand the central tolerance. So what's happening in the central tolerance is, once the B cell which are formed from the bone marrow is transferred to actually an area and start reacting to the self-antigen. So once the bone marrow cells are also reacting particularly towards the self-antigen, this cell has to undergo apoptosis. Otherwise, essentially these particular cell will produce more and more antibody in future, and therefore that's not good, and therefore these cells has to undergo apoptosis. The second part is what we'll study in the later point is called receptor editing, where the B cell receptor will be edited in such a way that they should not cross react with the self antigen. So if the B cell binds to the self antigen, there is a rearrangement in the light chain, and that can create an entirely different receptor which won't react to the similar kind of antigen. So this is what is called receptor editing, which we'll see a little later. And then it comes to peripheral tolerance, which is actually the tolerance at the level of selection that happens in the tissue level. So once the cells are escaped from the bone marrow, suppose there is no apoptosis happen to the cell in the bone marrow, the second line of control is at the level of tissue. And for example, the tissue here is spleen. So there could be a rearrangement of the light chain, receptor editing and stuff like that can lead to apoptosis of the cell which are reacting to the self antigen. The whole idea is the body maintains such an homeostasis that if at all the cells escape from the central tolerance, that should undergo the peripheral tolerance, where the cells are eliminated either by receptor editing or by clonal deletion. We will see how this critically synchronized so that the self-reacting antibodies are not produced in the body. But at all, after doing all this phenomena also, there could be a possibility that the body may react to the self-antigen, and people may suffer from diseases such as autoimmunity. And finally, we have germinal center selection, where we understood that the cells which are actually producing a very effective immune response, the B cell which are very effective in producing an antibody against an antigen, will be proliferated in a large quantity in order to produce a similar kind of cell type to clonal expansion, so that the same cell can destroy the antigen in future. So there could be more and more maturation of the B cell which are effective in nature, mature B cell population to be produced in large quantity in order to nullify the antigens. So this is what you call germinal center selection. So these are what you call germinal center selection. So in the next class, we'll try to understand the receptor editing and a little bit more about the tolerance and some more critical things about how these clonal aspect of a B cell and T cell population works. Thank you.